Thank you so much, everyone. Good morning, good afternoon, good evening, everyone. So this is my pleasure, great pleasure to welcome you to Bioturing webinar series, where AI meets biology. So our guest speaker today is Dr. Brian He. Brian did his undergrad at Stanford, right? And PhD at MIT. At some point, he spent a few years at Meta AI. And he's now a faculty at Stanford and investigator at Arc Institute. And Brian's work sits at the cutting edge of biology and machine learning. So Brian, so the reason why I invited you was of course, everyone here knows Transformer, but Evo 2 is mathematically very appealing to me. But Brian, Evo 2 feels like a moment where AI just doesn't just analyze biology, right? It begins to write it. And you have shown that it can predict variant effects. It can guide epigenomics design, or even generate entire genomes. Remarkably, it does all this without explicit labeling, right? So, under that power, there are several questions that I want to clarify. Do we really understand what Evo 2 understands? Is it learning biology the way that we describe it? Axon, motif, regulatory syntax? Or is it inventing its own internal grammar of life that just happened to work? And how do we, as scientists, best validate, use, or collaborate with a model this powerful? And how do I and other of my colleagues, as algorithmists, write new generations of genomics algorithm, like genome alignment, transcript alignment, genome assembly, or transcript assembly, with the availability of this new powerful model? So with that, I'm honored to hand the stage over to Dr. Brian He to tell us more.
Wow, thank you for such a kind introduction, and a lot of the questions were actually very well posed, and hopefully some answers at least, or very preliminary answers to those questions I'll address in the talk. So I'll go ahead and share my screen.
All right.
Okay, hopefully you can see this.
Yeah.
All right, so thank you, Sun, for the introduction, and I'm excited to share some of the work we've been doing on training biological foundation models from molecular to genome scale and across all domains of life. So I wanted to start out with this quote by Frances Arnold. This is actually from her Nobel lecture, where in 2019 she says, "Today we can, for all practical purposes, read, write, and edit any sequence of DNA, but we cannot compose it." So there's been tremendous advance in genome sequencing, genome synthesis and assembly, and even genome editing through technologies like CRISPR. But there's been much less progress, and it's been much harder to actually write and compose new DNA sequences, especially those that are substantially different from things that we can cut and paste from nature, as Frances Arnold says in this in her Nobel lecture. And fundamentally, this is because biology is written in a foreign language. So here is a cartoon illustration of the central dogma, as it's often called, of molecular biology, where primarily information flows from DNA to RNA to protein. And at each step in this information flow, biology is written in an unintuitive code, right? So I work regularly with protein sequences and DNA sequences and RNA sequences, and I probably still couldn't tell the difference between a completely random string of ACGT from the DNA sequence that was obtained from a living organism. But there are actually differences, right? It's not just random noise. There are patterns embedded in this code. So how do we actually extract these patterns? We were inspired by models that have made tremendous progress in learning higher level rules and emergent properties from raw sequences alone. And a lot of these models have been applied to human natural language. So they're trained on sequences of tokens where a token can be an English word, and they're trained with very simple training objectives like predicting the next token in a sequence. And for this first sequence at the top, to be or not to, probably everyone can predict the next word in the sequence. It would be B, especially if you've read Shakespeare. But then look at that second sequence where each token is a DNA base pair. We have GTG, CAT, CT. I don't think anyone could predict the next base pair in this sequence, but maybe to a machine learning model, these two tasks are relatively the same in terms of intuitive difficulty. And a key idea that underlies this whole project is that even though next word prediction, say in language, seems like a very simple task, if you train a model at on a sufficiently complicated data set, then next word prediction actually encompasses many very complex tasks within it. So for example, in this first sentence, in my free time, I like to code could be a valid completion, but in my free time, I like to banana would not be. So a good next token predictor would have to learn some understanding of grammar. But then in the second sentence, I went to the store to buy papaya, dragon fruit, and durian could be a valid completion of the sentence, but a squirrel would not be, even though it's a noun, it's not a fruit, and it's probably not something you could buy at a store. But then also a good next token predictor in language would have to learn facts about the world or how words are associated with human sentiment or translation between languages or spatial reasoning or math. So potentially lots of complex rules are encoded in next token prediction. And even though there's no explicit world model being learned by these large language models, the world has left its imprint on text from the internet. And so potentially you can learn facts about the world through sequence information alone. So what about in biology? So in biology, we usually like to think of genotypes as biological information, and these encode for phenotypes via some complex mechanism, right? And it's often these mechanisms and these phenotypes that we actually want to understand. But also, unfortunately, it's been very hard to collect enough data at the level of phenotype or mechanism beyond, very simple functions. But there's been a lot of advance in collecting data at the level of genotypes through next generation sequencing, for example. So what if we could actually learn information about phenotypes through genotypes alone? And why would this be possible? So in the same way that the world has left its imprint on text on the internet, the world has also left its imprint on genotype information through evolutionary selection. So, because there's information potentially about phenotypes through selection, we can potentially learn complex biological mechanisms through sequence variation. So, for example, if you learn which mutations are more likely to occur in some context, and you train a model on living organisms or functional organisms or healthy organisms, then those mutations might be correlated with health versus disease. If you learn how nucleotides come together to form codons and how those codons come together to form proteins, then you can learn some aspects of protein biochemistry. Or if you learn how RNA bases pair with each other, you can learn about RNA structure. Or even at a higher level, if you learn how genes come together, say to make a bacterial operon, you can learn some information about systems biology too. So this is a very appealing hypothesis. You could potentially learn very complex properties from sequence alone using very simple tasks. But is this actually the case? Like, is this actually a productive thing is this actually a productive paradigm that you can apply to biology? So first step that a lot of people in the bio-ML community have taken, including this is some of the work that I was doing when I was at FAIR in Facebook AI research, is training language models not on DNA, but on protein sequences. And these models are trained using very simple objectives again, for example, predicting the next amino acid in a sequence, or a related objective where you mask out or hide certain amino acids, and you fill in the blanks. So you predict the value of the amino acids given the amino acid context. And you train these models on hundreds of millions of protein sequences across basically all observed protein evolution. And the idea is these models learn rules as to how evolution has put amino acids together to form proteins, and these rules also encode complex biological information. And I won't go into protein language models too much into this talk, but suffice to say that these models have been very influential in protein science, and this is a snapshot of some of my work in this area. It's representative of a lot of different groups' work, but we can see that these language models learn rules of how of evolutionary dynamics and how proteins have evolved in the past that can be used to predict, say, the direction of evolution and historical trajectories. These models also learn an image of protein structure from sequence alone, and fundamentally this is because if two positions co-evolve with each other, then probably those two positions are in structural contact, because if you mutate one of the positions, you have to compensate with a mutation on the other position. So you can learn some aspect of protein structure from sequence co-evolution alone. And then you can use these rules to help guide evolution in the laboratory. So for example, we've shown that protein language models are useful in selecting a library for directed evolution. And more broadly, all of you are probably aware that machine learning has revolutionized the study of molecules. So for example, methods for protein structure prediction like AlphaFold or methods for de novo protein design were awarded the Nobel Prize in Chemistry last year. And this is definitely very well deserved. Molecules are very complex. We're also still very interested in molecules. But what was the key motivation in my mind behind the EVO work is we're also interested in how molecules come together to form complete organisms. But when you consider, say, even a very simple single-celled organism, this is orders of magnitude more complicated than any individual protein molecule. And we want to use machine learning to understand things at this level of complexity without losing molecular resolution. How do we even do that? How do we even collect enough training data to teach these models things about this scale of complexity? So fortunately, over the past 20 or 30 years, people have actually been sequencing the complete genomes of millions of organisms and depositing these genomes in publicly available databases. And in theory, embedded in this vast volume of DNA sequence data is all the information required to create complete organisms. But how do we actually extract this unstructured data from these sequences? So the around maybe a little bit more than a year ago, we trained a model that we called EVO, or for the purposes of this talk, EVO1. And this is a language model trained on prokaryotic genomes, so bacteria and archaea and phage. And fundamentally, this model, what it's doing is it's doing a very simple task. So it predicts, all this algorithm is doing is it predicts the likelihood of the next nucleotide in a sequence. So it takes in a sequence context and predicts the next base pair in a sequence. The model for doing this, though, is actually very large and it's trained on a lot of data. So EVO1 is trained on with 7 billion parameters in its neural network, and it's trained on 300 billion tokens spanning prokaryotic evolution. And we'll get in a second as to why scale is important for language modeling. But before that, the motivation for actually training models on DNA is that even basically because of the beauty of the central dogma, where you have the same information code coding for RNA and proteins as well, because you have the same information code underlying all three of these modalities, even though we're training a model only on DNA sequence, we could potentially get RNA language modeling and codon language modeling and protein language modeling and regulatory DNA language modeling for free out of this model as well. Even though we train on DNA, we can get multimodal information. But not only that, because we train this model to process a large segment of the genome, we not only, and EVO1's context length, the amount of the length of the sequence that it can process at one point in time is 100,000 base pairs, and the model is trained with single nucleotide resolution. So if at 100,000 base pair context, we not only get information about individual genes, but also how these genes come together to form multi-gene systems, and in some cases, even complete genomes. So going back to why we train these models at large scale, when we were training these models, we observed something that's been observed in other domains in AI, which are known as scaling laws. And this is the idea that as you increase the amount of compute used to train the model, then performance at the language modeling task, which is predicting the next token in a sequence, also gets better. So if you look at this plot on the right of the slide, on the x-axis is the flops used to train a model in terms of floating point operations. On the y-axis is the ability for the model to predict the next token. It's a metric called perplexity. Lower values of perplexity indicate better performance at predicting the next token in a sequence. And as we increase the amount of compute used to train the model's ability to predict the next base pair also improves with a pretty predictable trend. So scale is one dimension with which we can improve language modeling performance. The other dimension is architecture. So actually, the architecture that we use is different from standard transformers used to train other language models. And we observe that with the architecture that we use, this architecture called striped hyena, we actually have favorable scaling performance to more traditional architectures. So this motivates training multi-billion parameter models, but what actually can these models do? So we roughly think of the downstream applications of these models in terms of two broader categories. One is prediction tasks, and the other category is generative tasks or design tasks. So I'll talk about one prediction task that we did that it's probably one of my favorite things that we did with Evo 1. So for this prediction task, we are going to see what happens when we introduce a genetic perturbation to a sequence and then evaluate Evo's likelihoods on that perturbed or mutated sequence. And the perturbations that we're going to make is we're going to introduce a premature stop codon insertion into each of the coding sequences in a long segment of DNA. In some cases, this sequence of DNA is so long it contains the entire genome of the organism for some phage, for example, with very short genomes. And when we do this, we actually observe that when we introduce a premature stop codon into the sequence of an essential gene, Evo's likelihoods go down actually by a lot on average. But when we put a premature stop codon insertion into a non-essential gene, Evo's likelihoods don't change all that much, or they change much less. So the model understands which genes in a genome are more sensitive or not to these very bad premature stop codon insertions. If you actually made them in the organism, they would knock out the gene by ensuring that it doesn't code for protein. And in some cases, this predictive performance even is so good it even almost recapitulates the data from experimental gene essentiality studies. So for example, in this data set, this is a genome-wide CRISPR-I screen in lambda phage. The zero-shot Evo likelihoods almost perfectly predict gene essentiality in this organism. And I'm just highlighting that Evo had no explicit training on what genes were essential or not. It just learns which genes are more tolerant of these bad mutations, and so it can perform zero-shot gene essentiality prediction. So the next category of tasks that we did were design tasks or generative tasks. And for this, we wanted to design a very complex system, actually, with multi-modality. So this is a CRISPR-Cas system for doing genome editing. It actually involves a very complex protein. Cas9 is almost 1,400 amino acids, very long, bound to a guide RNA that itself is composed of two different non-coding RNAs, a tracer RNA and a CRISPR RNA. And in prokaryotic systems, these sequences are often directly next to each other on a CRISPR-Cas operon. So we fine-tuned Evo on natural examples of CRISPR-Cas systems and then generated that with that fine-tuned model. And one of the sequences that this model generated, this locus that we called EvoCas9-1, actually had a full-length Cas9, a detectable tracer RNA and CRISPR array, also other proteins that often co-occur with Cas9, like Cas1 and Cas2. When we put the sequence of Cas9, of EvoCas9 and the tracer RNA and CRISPR RNA into AlphaFold 3, the predicted structure resembles that of known CRISPR-Cas complexes. And then we can actually synthesize this DNA and test it in the lab. And we show that in vitro, the EvoCas9 plus EvoCas9 guide RNA complex has comparable in vitro cleavage to SpCas9, which is this state-of-the-art genome editing tool. But also, the generated molecules have less than 80% sequence identity to any Cas9 we could find in nature, 73% identity to SpCas9. So this is actually novel enough to break any patents around CRISPR-Cas. And so at least these molecules are somewhat novel in the eyes of the patent office, for example.
Wow.
We wanted to push the novelty threshold even more. And for this, we leveraged the fact that Evo can actually have a multi-gene understanding of the genome. And you can prompt Evo with genes that often co-occur with other genes in the genome and use it to, say, autocomplete bacterial gene clusters or operons. And there are some bacterial gene clusters that often encode de novo genes. So, for example, these are often gene clusters associated with the bacteriophage arms race. And if you have a very highly conserved gene there, it really disadvantages you in the arms race. You want a source of evolutionary novelty in these regions. And so we then used Evo to prompt the model with what's called an anti-CRISPR associated protein to generate an anti-CRISPR protein. These are proteins that inhibit Cas9, inhibit CRISPR. And the anti-CRISPR proteins in nature are often are hypothesized to arise via de novo gene birth. When we actually test these Evo-generated anti-CRISPRs in the lab, we can confirm that they do have anti-CRISPR activity. But also, when we look at the amount of novelty of these generated proteins, they basically have almost no, well, they have no significant alignments to any protein in the training data, any protein that we could find in nature, actually. And we do various analyses and can show that the level of novelty of these proteins is even comparable to de novo proteins designed by David Baker. And this shows, in my mind, that these models are not just memorizing or regurgitating their training data, but they do actually can be quite creative and generalize non-trivially beyond the sequence space in their training data.
Yeah.
We also then take this a step further and use this technique to generate 100 billion base pairs of AI-generated genomic sequence spanning many putative functions beyond CRISPR or anti-CRISPR. And then the most forward-looking science fiction part of the EVO1 paper was we then used the model to generate sequence at 1 million base pairs. And we thought these sequences would be totally unreasonable, but when we actually analyzed these sequences with various metrics that people use to analyze the validity of genomes, they were actually somewhat reasonable according to different metrics. So for example, the coding density of these sequences is similar to what you would expect in nature. The predicted ORFs also have predicted secondary structure when we fold them. And then we also found tRNAs, so non-coding RNAs on these sequences that cover all 20 canonical amino acids, for example. So maybe kind of we liken this to the first versions of image generation models that would generate somewhat coherent, but maybe blurry pictures of objects. In this case, this first version of whole genome generation is somewhat reasonable, but maybe also room for improvement. And then what we wanted to do after this first version of Evo is also apply it to eukaryotic genomes. So for example, the genomes of plants and animals Humans are eukaryotes, so if we want these models to be useful for human health, it also would be nice to apply them to eukaryotic genetics and genomics. The problem with eukaryotic genomes is that these genomes are much more complex. They're much longer than prokaryotic genomes. They have more complex genetic architecture. There's splicing and lots of non-coding sequence and lots of repetitive elements. So we needed to do a much better job also at data processing and data engineering before training these models. And the resulting model that we trained is a model we put out a few months ago now that's called EVO2. And this is a model that is trained on all domains of life. So it actually is trained on more prokaryotic data than EVO1, but it also is trained on eukaryotic data spanning plants and animals and insects and non-human primates and humans. So this is a UMAP plot. Each dot corresponds to a different organism trained in the EVO2 training data. And we spent a lot of effort, including from these two bioinformaticians, Matthew Durant and Garrick Brixey, on actually curating this data so that we were focusing a lot of the models learning on functional sequence. We also wanted to extend the context length of this model beyond 100,000 base pairs because eukaryotic genomes are much longer. So with EVO2, we extend to 1,000,000 base pair context while preserving single nucleotide resolution, so we can still understand things at the level of genes and proteins and non-coding RNA. But now we can also place these sequences in the context of minimal prokaryotic genomes or yeast chromosomes or even human TADs. We also scaled the model in its parameters up to 40 billion parameters. This actually required a lot of engineering. It required a lot of compute. We partnered with NVIDIA, who supplied the compute for this project, so we're very grateful to them for this. And at the amount of data used to train the model, we trained this model at 40 billion parameters on close to 9 trillion tokens. This actually, by floating point operations, so by the scale of compute, actually makes EVO2 the largest model ever trained on biological sequence data. And also, because we make this model what's called fully open, so we not only open source the model weights and the code for doing inference with the model or running the model, we also open source the training data and the code for training the model. So this is a fully open model. This actually, by compute flops, makes EVO2 the largest fully open model across all modalities, including text and code and images. So the part of the reason we were able to achieve such long context lengths with 40 billion at such large parameter sizes, 40 billion parameters, is because we did a lot of architectural modifications, lots of constant time improvements, also some asymptotic improvements in the architecture using an updated architecture called Stride-Tayuna 2. So again, what can you actually do with this very large model? So the model, the way I like to think of it, one way I like to think of it is it's seen a lot of DNA base pairs from living organisms. So it's learned the rules of basically which mutations or which sequences are compatible with life. So if you have, say, variation in the genome of an organism, then mutations with a high likelihood under EVO, or EVO2 in this case, would probably correlate with health or function or compatibility with life. But if a mutation has low likelihood under the model, then potentially this mutation is correlated with incompatibility with life or with function and therefore pathogenicity and disease. So in the same way that we scored mutations to prokaryotic genomes for gene essentiality, we scored mutations to human disease genes for their pathogenicity and leading to loss of function. So as one example, we applied EVO2 to scoring variants of BRCA1 that were experimentally determined to lead to either functional or intermediate function or just complete loss of function. BRCA1 is, of course, a gene that is often implicated in breast cancer, and it also has human disease relevance. So actually, when we benchmarked EVO1's zero-shot likelihoods at this task, it has very competitive prediction of single nucleotide variants in coding regions for their pathogenicity, almost on par with state-of-the-art models. It's also particularly good at mutations in non-coding parts of the genome. So uh, uh, it's state-of-the-art in non-coding regions. It's also, the data is not shown here, the model is also state-of-the-art in mutations that are not single nucleotide variants. So insertions or deletions or small structural variation, things like that, the model is state-of-the-art there. And then I just wanted to highlight that this is what's called zero-shot performance. So the model has actually only been trained on evolutionary information from many species. It's not been specialized to human genetics. It's seen no human-specific information. So as a follow-up study, we then fine-tuned a model on top of EVO2's representations on some task-specific data relevant to BRCA1. And when we do this, we do actually have, again, now a state-of-the-art performance on coding mutations for this task. Moving on, another set of prediction tasks we wanted to see is how well can we interpret this model? So, contrary to the prevailing notions of neural networks as black boxes, mathematically, they're actually not black box functions. You can pass gradients through them. You can inspect all of the model's parameters. And there's been a very productive line of work in doing this for models of natural language. So for example, this is a interpretability study done by a company called Anthropic on a model called Claude. And they found internal representations learned by Claude, this model, in this case, that only activate when you provide an input related to the Golden Gate Bridge. And actually, they found that this feature actually activates not only on English text of the Golden Gate Bridge, but also across multiple languages too. So inspired by this, we also wanted to conduct an interpretability study on the internal representations learned by EVO2. And the idea underlying this kind of interpretability is that these concepts that make sense to us as humans are present in the network, but maybe they're in some dense combination in the raw representations. So you want to kind of disentangle those concepts from each other by training a sparse representation or a sparse, and so we do this with a sparse autoencoder and interpret the features of this sparse autoencoder for congruence with known human features of the underlying biology. When we do this, we actually find features that activate only on certain genetic regions and can actually be used to do a kind of genome annotation or segmentation. So for example, we find features that activate only in regions of non-coding RNAs. We find features that activate on protein level regions, so on alpha helices or on beta sheets. We also find features that activate in regions corresponding to transcription factor binding motifs. So we have DNA, RNA, and protein level features learned by the model. The model also learns features associated with higher level genomic architecture. So for example, annotating the start and end of introns and exons and coding sequences. We, as a kind of toy task, apply this to annotating the transcriptome sequence in the woolly mammoth genome, a task which does require computational prediction until we resurrect the woolly mammoth and do RNA-seq on it, for example. And then another thing we found is that this kind of interpretability finds errors in computer code and genetic code. So for example, the team from Anthropic found a feature that activated when you have a bug in your Python code. So if you have a typo in your Python script, this feature activates. In our interpretability study, we found a feature that only activates in frame shift mutations in eukaryotic sequences. And it actually doesn't matter the sequence composition of the frame shift. the model identifies any kind of frame shift as a frame shift. And in this way, the model does, we think, learn kind of rules that are almost kind of invariant to actual literal sequence motifs. It's not just trivially looking up, not constructing a lookup table of all possible frame shifts and all possible genes. And in this way, the model is actually finding errors in genetic code. The final class of tasks we apply this model to, and again, these are a little bit more forward-looking, but potentially an emerging area, is in using this class of models to generate things at genome scale or at the scale of very long DNA sequences. So for example, as one computational task, we wanted to see how well the model could generate, say, complete human mitochondrial genomes or minimal prokaryotic genomes or yeast chromosomes and see how coherent these generations are in comparison to previous versions, basically Evo 1, a previous model for doing large scale or very long sequence generation.
Mm.
So, for example, the model does seem to have much greater coherence, especially with scale. So, for example, in this generation task, we wanted to generate variations of the human mitochondrial genome. And actually, these generated variations have all of the right components that you would expect to go in a mitochondria. And here's an AlphaFold 3 structure prediction of an Evo-generated electron transport chain, for example. It does form a multimer complex that with the expected structure. And then comparing this to Evo 1, the coherence at prokaryotic genome scale is actually much better for Evo 2. This is according to a number of metrics, including coding density, including how many conserved genes we can find on these genomes and the naturalness of these genomes, and even the structure prediction confidence, things like that. And for the first time, we can also generate genome scale sequences with hallmarks of eukaryotic genome architecture. So for example, in these yeast chromosome scale generations, there are introns and exons and the right promoter sequences from yeast chromosomes. All of this generation is what's called unconditional generation in machine learning. We basically don't tell Evo to specifically generate something, right? We just generate freely from, say, a yeast chromosome or prokaryotic genome. So for our last task, what we wanted to do is actually demonstrate how you would be able to control the outputs of Evo 2. And for this, we leverage a line of work that's been referred to as inference time compute and guided generation. And basically, the idea is you couple a generative model with a separate scoring function or reward function that can steer the outputs of the model to a very specific task of interest. So for this task, we have the somewhat toy example, but kind of fun and whimsical, of encoding Morse code into mammalian genome sequence. And the way we do that is by the way we encode Morse code is in the chromatin structure of this de novo DNA sequence, where a narrow peak corresponds to a dot, a wide peak corresponds to a dash, and heterochromatin makes the spaces between the peaks. Just to demonstrate that we can control both the length and the location of these peaks in de novo DNA sequence. To do this, because Evo 2 does not have an explicit understanding of chromatin accessibility, we couple it with these models like Informer and Borzoi that do and can predict chromatin patterns from sequence. And we do what's called a beam search algorithm. So we don't generate the whole sequence all at once. We actually generate the sequence partially in chunks. And as each chunk gets generated, we score that chunk based on how well or how compatible it is with a chromatin profile that we want to explicitly design. We accept sequences that fit that profile, we reject sequences that don't fit that profile, then append that chunk to the prompt and just generate the next part of the sequence. So here is a GIF showing one design trajectory. The x-axis is the position into the genome. The y-axis is the predicted chromatin accessibility. The bold yellow line is the Morse code pattern that we actually want to design. And the light design is the light blue or gray and white lines are the predicted accessibility profiles of the de novo sequence that we're designing. And as it goes from left to right, that each frame is basically a new chunk generated by EVO2 that then gets accepted by the model and goes into the final design. And you can kind of see how this works. The model is basically writing chromatin accessibility from left to right in real time, where we've actually synthesized this DNA and have inserted it into mouse ES cells and are doing the attack-seq to confirm that these designs have the desired chromatin architecture. So stay tuned for results on that. We're actually experimentally validating these designs.
Is it confirmed yet or not yet?
I don't want to spoil the surprise. So I don't want to confirm or deny anything. Yeah. But in general, it might be a tractable task. I don't know. In general, we do think that this we do think that this paradigm for generating a sequence with a very capable foundation model or generative model, and then coupling it with a reward function or a scoring function could be useful beyond this task of chromatin architecture, epigenome structure design. For example, designing desired protein structures, or if you have any function that predicts a function or sequence to function predictor that predicts a function of interest from a sequence, you can couple that with a generative model to steer that model to design sequences of desired function. And because this model has learned kind of the sequence landscape of biology, you have a much better prior and a much higher likelihood of downstream success. This general paradigm actually has been very useful in protein design with protein generative models like the protein language models. But now that we have genome language models, we think that this paradigm is also applicable to, say, designing more complex genomic sequences. Okay, so in summary, we have seen a single basically a single model or a single modeling paradigm that's capable of prediction and design tasks at the scale from genes to entire genomes. So for example, we've seen how the model can be used to score single nucleotide variants that have an impact on gene function, like BRCA1 function. We've seen how the model can predict genome segmentation and which regions of the genome encode for different things. And also, we've seen how the model can predict how mutations to genes affect whole organism fitness and predict gene essentiality in the context of a whole genome or a whole organism. We've also seen how the model can design individual genes like anti-CRISPR proteins or multi-gene systems like these protein RNA complexes that like CRISPR-Cas. And potentially these models can also be used to do design tasks at the scale of very long sequences, even potentially in the future, minimal genomes. So going back to this quote, there's been tremendous advances, right, in DNA reading, writing, and editing through sequencing, synthesis and assembly, and genome editing. And where we see this research as fitting in, as we're basically learning the rules from how evolution has composed sequences throughout all of evolutionary history. And we're using that information and distilling it into a model to enable genome compose as well. So this could be the last step in the loop. We have read, write, edit, and compose. And together, this suite of technologies will finally enable genome design. So that concludes a lot of the bulk of what I wanted to present today. We have made a lot of these details. Well, I've glossed over a lot of information for the sake of time, but we've made a lot of these details, or all the details available in our publications. So for EVO1, these are the references, the preprints and publications on this work. And we've also made the models publicly available And the code for running the model is publicly available at this GitHub link if you're interested. These also are the references for Evo2, two preprints, one describing the model and its broader training and biological use cases. We also have a machine learning supplement to the paper that goes into all the gory details of training large models at long context at scale. And so if people are
Yeah.
interested in that, you can take a look at this archive paper. And also the model, the code for running the model, the code for training the model, and the training data are linked to at this GitHub. Finally, this is work that is done across a number of different institutions spanning many different areas of expertise. So we have people working on machine learning engineering and scale. We have people working on computational biology and evals, on interpretability, on experimental biology, and actually validating that the model works in the wet lab. Particularly want to call out, Anthony Costa, Mingyu Liu, and Kimberly Powell at NVIDIA, Dave Burke, Hani Goodarzi, and Patrick Su at ARC as the senior collaborators and leads for this project alongside me. And this is work done across ARC Institute, Stanford, and NVIDIA, and other academic and startup institutions as well. So I represent a very large and talented team with this. And finally, thank you all for your attention. Happy to take any questions now. I think we still have, actually some time to talk and discuss and
Thanks
answer
so much.
questions.
Thanks so much, Brian. This is amazing work. This is amazing work. And how
Yeah,
many
thank you.
GPU did you train on that?
Yeah, so the the big scale up, the Evo 2 40B training run ran on basically a compute allocation of around on 2048 GPUs for around 3.5 months. So it was a
Mm.
fairly large scale up.
Well, okay. So let me go through. We have a series of a lot of questions, Brian.
Great.
First question is like, which downstream tasks can you list that Evo 2 can be useful for so that, scientists can start thinking on how to use it?
Yeah, I think so basically
Predict
there's
mutation.
exactly so I think variant effects already, especially especially for non-coding variant effects and for indels, like not single nucleotide variants, the model's already state of the art and can be directly useful. Now, as seen in this downstream test as well, the embeddings of the model are also useful when even if the model is not zero shot state of the art, you can fine tune the model or train a supervised model on the embeddings of Evo 2 on your task of interest. And then because you're starting off at a really strong foundation already, the zero shot performance is pretty good. Then a little bit of supervised training with much less training data than would be required on an if you're training a model completely de novo,
Right.
would hopefully get you to state-of-the-art performance. So in terms of prediction tasks, either using the direct likelihood outputs of the model zero shot as like a evolutionary prior that's predictive of your function of interest, or using the internal representations and the embeddings of the model in a workflow. That
Right.
those are a general way of using the model. And then also for generative tasks, uh, we have various ways. This is more described in the papers. We've done a lot. We've actually put out a lot of work showing that you can prompt engineer these models in various ways. And depending on the DNA sequence you use to prompt the model, you can actually get very interesting designs in the output. So for protein engineering tasks or de novo design tasks and proteins, and potentially we're working on this now, more complicated multi-gene systems, the model will also be useful as a generative model.
Right. Thanks so much, Brian. Another questions from a colleague of mine asking, um, the mutations, of course, it depends on it's context dependent, of course.
Mm.
You have mutations, but maybe surrounding that, you have different mutations that that can change the effect of the current mutations. Are you taking the context around it for predicting?
Yes. I think there's maybe two aspects of the question that are both good to answer. The first is the first is yes. When we actually do this variant effect like likelihood scoring, we we need to provide the model with some genome context, right? We don't just give it the single variant that's different. We see performance gains up through around 10 kb in this variant effect prediction task, like 8 kb to 10 kb. There, the genomic context still improves performance. Beyond that, it kind of levels off and plateaus. And that kind of makes sense. Lots of these variant effects are just very local, so you might need to understand the
Right.
genic region or the gene that this variant is embedded in order to get some, insight into its function. Then there's a second question, which is, does the model learn like nonlinear interactions with other parts of the sequence in its context? Uh, I think this is much harder. There's maybe some evidence of this, but very weak. I do think like a good what we would maybe want to focus on in future model development in this line of work is actually trying to capture more of these nonlinear or distal effects. But at least in terms of these local effects and leveraging the context, the model already does pretty well and is state of the art in certain tasks.
Yeah. Another questions regarding the essential the predictions. Have you conduct this analysis on DepMap, the cancer gene dependency data? So the colleagues was curious if EVA2 could predict rigorously by capturing cell type and tissue specificity of cancers.
Yes. We don't. So we don't actually. We don't have this in the paper, and the model does not learn cell type specificity or tissue specificity. If you wanted to incorporate that, I think you'd just have to augment the model with additional supervised training or something. We've done a little bit of work looking at depth map gene essentiality. We actually see that the gene essentiality performance improves the more the gene essential more the more cell types or tissues that a gene is essential in, the better the gene essentiality prediction performance is. Or maybe a equivalent way of phrasing this is like if a gene has a more conserved function across many different tissues in terms of its essentiality, then the model does understand its essentiality better. And in terms of data, we actually have shown this for this essentiality screen in long non-coding RNA. So in the EVA2 paper, we have long non-coding RNA essentiality and we're working on the depth map essentiality analysis as well. But that maybe gives an insight into what the model is learning. This model has learned information across multiple species. So things that are more, conserved in human essentiality across different tissues and cell types, that will be closer to the kinds of mutations that are bad and stuff that the model has learned in its training data. But the model has no explicit training on tissue specificity, for example.
Right, right, right. And so cell types and tissues is a little bit. So you start from the same DNA here, right? Um,
Exactly.
the questions from Adam. Prompting, prompting. How do you prompt? How do you prompt? Should you initiate with some sequence? And how are you thinking of integrating the human language into this?
Yes. Right now, there's actually there's kind of natural language prompts because we have a species tag that the model's directly trained on that encodes like the phylogenetic string of the species. So you can actually prompt with the species tag and generate DNA. For this generation task, we actually fine-tune Evo1 on CRISPR-Cas sequences with a special token in the fine-tuning data set so that when it sees the special token, it knows now I need to generate a CRISPR-Cas sequence. And then we prompt just with that special token, and then it generates the CRISPR-Cas sequence. And then we've shown that the model can also, you can prompt it with a gene, and then just generate the gene next to it. And this is useful for prokaryotic design tasks in which the architecture of prokaryotic genomes has multiple functionally related genes next to each other, and the actual function of these genes is somewhat predictable given its neighbors. So we can leverage this property to design specific functions that we want. These are basically creative ways of prompting the model with DNA or with tags that are associated with DNA. I think what we are actually interested in, but not for this model, for future versions of the model is, yeah, what would it look like to prompt the model with natural language and have it generate a DNA sequence that would have the desired function. I think that is maybe a much more complex task than image generation, for example.
Yeah, and how about the connecting with human language? Anything?
I think, yeah, like the current version of the model are not trained on human language, but yeah, exactly. What we're very interested in what it would look like if you wanted to steer, say, like steer Evo's generations,
Yeah.
like maybe using this paradigm where you steer Evo's generations using a natural language reward or scoring function or something.
But by steering, are you calculating the reward function on the fly and feedback it, provide feedback immediately, and it generate new ones?
Yeah, so for what we do now, that we have no additional fine tuning or we're not updating the generative model. It's totally inference time.
Ah,
And that's the sense in which the feedback is occurring. It's just rejection sampling. But there are ways of actually, incorporating this experimental or incorporating this reward, this in silico feedback into the generative model, like via uh, reinforcement learning inspired fine-tuning objectives and things like that. This is something we're also very interested in.
right, right. Thanks so much. Have you tested on some tasks for predicting enhancer, like regulatory elements?
Yeah, well, we actually have some very interesting results on like this TF motif
Mm.
prediction, uh, mech result, and potentially some of these TF motif features might actually be quite good, even in comparison with traditional methods for motif discovery, that this is still a work in progress. And
Yeah,
I can imagine something being the same for enhancer identification as well. A nice thing about this kind of genome segmentation with the model is the model's actually learned these regions and these features autonomously. So this is
well,
completely unsupervised learning. So the model, and this goes back to the initial kind of the actual introduction, that some of these features that the model has learned autonomously that we can interpret do correspond to known biological concepts that we can interpret. There are many features where we just don't know how these genome regions actually relate to each other, or if there is some unknown function that we have not yet discovered. And if we have a feature that has, if a feature that has, high precision or high recall of a TF motif, but then there are, things that are not known to be TF motifs, but that have high activations under this model, maybe they do actually have some biological basis. I think this is this kind of work would be really interesting for discovery. Um,
right, Brian. There are a lot of questions. There are 34 more open questions, but we are running out of time now. So I will send you the list of all the questions so that we can distribute it to the audience. Brian, thank you so much for this amazing talk. And
Thank you so much for having me. This is fun, and the questions were great. And thank you again, Simon. So,
all right, thank you so much, Brian.
all right,
Have,
bye.
have a great day. Bye
You
bye.
too.